So welcome to everybody. We start this session on tolerance induction in myasthenia gravis. We have two presentations, and the first speaker will be Konstantinos Lazaridis from the Hellenic Pasteur in Greece, and the title of his presentation is Preclinical Study of an Antigen-Specific Therapy for the Treatment of Myasthenia Gravis.
Thank you, Emilia. Let's see how this works. Okay, thank you. And thanks to the organizers for the invitation and the opportunity to present our work. Thank you very much. I will talk to you about our efforts to develop an antigen-specific therapy for MG. Yeah. So some of you may have seen this before, so I won't go into it in too much detail, but just to highlight that we are working on AChR-MG, being the most prevalent form of the disease so far. So an array of therapies are already available for the management of MG. This list is by no means comprehensive, but there's several more options available. So why do we need another approach? Well, none of these are antigen specific. So they can have reduced efficiency, at least for some patients, or they can be accompanied by side effects. So I think it's important there's a lot of room for improvement there. The ideal therapeutic approach should only target the pathogenic cells and components of the immune system involved in the pathogenesis, and leave all the other cells and components free to do their business. We know that exposure to antigen can induce tolerance, and in fact, in MG, several groups have long shown that oral or nasal administration of acetylcholine receptor can either protect from EAMG induction in animal models or treat ongoing experimental EAMG. So we're going to look into this a bit more. This is the acetylcholine receptor. The subunits is composed of 5 subunits, which have the extracellular domains, which contain the pathogenic, the disease-relevant epitopes. And we have previously optimized expression of each of the extracellular domains of each of the subunits as soluble proteins with good near-native conformation in the yeast Pichia pastoris. So we will focus on the alpha-1 ECD for these studies, being the most immunogenic in rats, containing the MIR, and maybe being the most pathogenic in human MG as well. We are using an EAMG model in rats induced by immunization with the human alpha-1 ECD. And we will use this model to investigate the therapeutic potential of alpha-1 administration via different routes, and we will explore the effect of dosing and timing of administration and so on. So I already mentioned that we know that nasal administration has been shown to be effective in the treatment of EAMG, but when we compared intranasal to intravenous administration in our animal model, we saw that it was much more efficient at treatment of EAMG. And these results are from treatment started 7 days after disease induction. I should say that all of the experiments that I will show you involve a treatment regimen involving 12 consecutive daily administrations of antigen. Similarly, when we compared IV to IP or subcutaneous administration, again, IV was more efficient at treating EAMG. And to verify that this is an antigen-specific observation and not some non-specific effect that we are seeing, we compared administration of human alpha-1 ECD to ovalbumin and to MuSK, which of course is also MG-related, but has no homology to the acetylcholine receptor protein. And we saw that only administration of the alpha-1 ECD had a therapeutic potential. All right, so looking a bit in more detail with respect to the time of the administration. First of all, treatment starting 7 days after immunization, we saw a dose-dependent response. The more protein we administered, the better the results. Similarly, when we looked at 21 days after immunization, again, a dose-dependent response, but in this case, we needed more protein to have a similar effect. And again, when we started treatment on day 40, again, a dose-dependent response, but even more protein being required for a similar effect. And the data that we're showing here in the classical EAMG score are always corroborated by measurements of changes in body weight of the animals, acetylcholine receptor muscle content, and compound muscle action potential measurements. So we know that more is better, more protein is better, but how about the frequency of administration? So how does the total amount of antigen relate to the number of doses? Could, in other words, knowing that we need more protein, could I give a big amount of protein in 1 or 2 injections and be done with it? Well, to do this, we gave the same total amount of antigen, in this case, 1200 micrograms, divided over different number of doses, 3, 6, and 12. And what we saw was that if in the rats that received fewer injections of higher antigen dose, the effect was not as profound, and the effect was much better when smaller doses were given over many more injections. So more is better, not only in terms of the amount of antigen, but also in terms of the administrations required. We need several administrations to have the optimal effect. And this led us to investigate the fate of the protein following IV administration. And to do this, we used radio-labeled protein, injected IV, and then taking serum samples over a period of 6 hours to measure the amount of protein that we see. And we also collected at the end of those 6 hours several major organs to study the distribution of the protein. So what we saw was that there's a rapid decrease of the protein concentration, especially in the first few minutes following the injection. But this was not really influenced by whether we were looking at doing the experiment in non-immunized rats or rats immunized 21 or 40 days before the experiment. And the organ distribution, perhaps not surprisingly, most of the protein is sequestered in the liver and some appreciable amount in kidneys and spleen. But again, the image was, the effect was the same, irrespective of the experimental animal group that we studied. Next, we wanted to compare the efficiency of alpha-1 administration with a corticosteroid to see how it compares to say, a current therapeutic. And to this end, we used methylprednisolone given IP. We studied the response at 7 and 40 days, and we saw that alpha-1 administration was more efficient at treating disease, whereas methylprednisolone, the rats that were given methylprednisolone presented with symptoms after the treatment was terminated. Another aspect that speaks to the applicability of the approach is the potential to use E. coli produced protein, because it's usually, probably easier to produce it in larger quantities and easier. So we compared protein made in yeast and E. coli at, again, 2 different time points, 21 and 40 days. And we saw that they didn't have any discernible differences in the observed effect. Which could also hint as to the involvement of the structure of the antigen and the glycosylation of the antigen, the mechanism of action. They might not have a very significant role to play there. So what is the mechanism of action? Well, we started by looking at the antigen concentrations in serum samples from different time points following induction and treatment of rats. And here, surprisingly, we saw that especially for the rat groups that received treatment 21 and 40 days after immunization, there was an increase in the antibody titers, both for rat AChR antibodies and alpha-1 antibodies. But these titers did not correlate at all with emergence or severity of the symptoms. So there's clearly a lot we still don't understand about the nature of these antibodies. I don't have any more answers, unfortunately, to give you on this, but we are looking into aspects of these antibodies to understand their nature better in terms of their pathogenicity or potential pathogenicity. So, for example, if they can induce antigenic modulation or complement activation, or if they can passively transfer disease to other rats. Looking further into the mechanism of action, we studied proliferative responses from cells collected from spleens and draining lymph nodes from rats following treatment. And we saw that there's a decrease in the proliferation observed in cells obtained from treated rats. And similarly, looking at cytokine profiles, we saw that cells stimulated in vitro from rats that had received treatment had decreased expression of interferon gamma, production of interferon gamma and IL-17, and an increasing production of IL-10. And these were all experiments that were done in rats treated on day 7 only. So there's still a lot to be done. We still need to explore treatment, the response to treatment at later time points. I think it's very important to understand what's going on there as well, but also look into these groups in more detail. But this is where we stand at the moment. Briefly to summarize, we have seen that intravenous administration of alpha-1 ECD can be very efficient at treating experimental MG in our model. We have characterized several aspects of that affect the efficiency of the treatment. We have seen that alpha-1 administration was more efficient than a corticosteroid, and we are starting to investigate the mechanism of action of the disease. This is where a lot of the work remains to be done. We certainly need to characterize the antibodies that are produced following treatment, especially in the rats that are treated at later time points after disease, but also to identify the specific cell subpopulations and cytokines that are involved and the key players that are involved in the observed phenomenon. Equally important to try to improve the characteristics of the antigen to potentially increase its serum half-life, which might improve its potency. Or to use encapsulation into nanoparticles, which would not only protect it, but open up a whole host of possibilities in terms of what you can do, and such as improved targeting, for example, and so on. All of these, though, I think are very important, not only to understand how tolerance works better and hopefully allow its application. It might help understand some aspects of disease pathogenesis as well. And very importantly, allow us to design even better therapeutics for MG, more targeted. Just a few seconds to acknowledge all the people who have made this work possible. First of all, my team from the Hellenic Pasteur Institute, and especially Vicky and Eleni, who have performed the majority of the bulk of the work that I showed here today. All the people from the animal research unit, and our good collaborators from Toleranzia, who've also provided the E. coli express protein we used in some of the experiments. And thank you all. Thank you very much for listening.
And the second presenter is Debbie Reuveni from the Weizmann Institute of Science, Israel. And the title of her presentation is Oral Tolerance of MuSK-MG.
Thank you for inviting me. It is a great pleasure to be here today to be able to present to you one of my projects during my postdoc, which I perform at the lab of Sarah Fuchs and Miri Sorojon at the Weizmann Institute of Science. So the topic of my talk is, as said, induction of suppression of the MuSK myasthenia gravis by induction of oral tolerance. During my postdoc, Miri Sorojon passed away, so I would like to take this opportunity to dedicate my talk in her memory. So let's start, but I don't know how to use it. Okay, so I would like to take you back to the year of 1672, when Dr. Thomas Willis noticed these patients that were fine in the morning, but fatigued towards noon and were unable to speak for a long time. And this is actually the first description that we have, one of the first descriptions that we know of myasthenia gravis. And when we take a look on the history of myasthenia gravis that we heard yesterday from this beautiful talk of Angela, we can divide these times into 4 periods of times, in which at the beginning more and more observation of these patients were accumulated, so all the characteristics of myasthenia gravis were defined. Later on, acetylcholine was discovered to be the transmitter at the neuromuscular junction. The most changing times, in my opinion, are in the 60s to the 90s, where a lot of things happens, which we can say it's a revolutionary era for myasthenia gravis. And one of the main discoveries was the isolation of the acetylcholine receptor, which opened up new opportunities for researchers to study myasthenia gravis in experimental models. Also, the discovery of these antibodies against the AChR led to the recognition that myasthenia gravis is an autoimmune disease. And some treatment modalities that put into use, such as IVIG, plasma exchange, and the use of corticosteroids, changed dramatically the prognosis of these patients. So the last period of times from the 90s and so on, it's characterized by immunological progress that led to the discovery of these anti-MuSK antibodies. So it took 350 years since myasthenia gravis was first described until the recognition and definition of this subpopulation of patients that are MuSK-MG. So we heard a lot about MuSK, so I will go briefly. It was originally described in the electric organ of Torpedo californica as a tyrosine kinase receptor, and it was later on isolated from denervated mouse muscles, so it was termed muscle-specific kinase. It is a 100 kilodalton transmembrane tyrosine kinase receptor, which have a terminal extracellular domain carrying 3 Ig-like domain and 1 cysteine-rich domain, a short transmembrane domain, and the C-terminal cytoplasmatic domain carrying the catalytic activity. So although MuSK shares similar features with other RTKs known, its mode of activation is distinct in a way it requires 3 additional proteins for full activation. It requires agrin, which is derived from the motor neuron, the LRP4 located at the muscle membrane, and the DOX7, which is expressed in the cytoplasm of myofibers. So the role of MuSK is actually in determining the efficacy at the neuromuscular junction. And it does it by associating with the LRP4 via the Ig1 domain. This tetramer complex is then phosphorylated by agrin, resulting in the recruitment of DOX7, which enhance the activation and phosphorylation in MuSK, causing this post-synapse differentiation, resulting in the AChR clustering. So what we know happens in MuSK myasthenia gravis patients is that the presence of these anti-MuSK specific antibodies that bind specifically to the Ig1 domain blocks the association between LRP4 and MuSK, thus resulting in this impaired signaling through MuSK, eventually leading to the decrease in the AChR. So this neuromuscular transmission is impaired. As we all know, the antibodies present in these patients belong to the Ig4 family that are unable to activate the C complement system and immune cells. So MuSK-MG patients are reported in about 5 to 8% of the patients, and its prevalence varies among countries and ethnic groups, with higher percentage in Southern Europe. It predominantly affects women and with a peak of incidence in the late decade of life, but rarely occurs after the 70 years of age. And clinically, these patients are different as they suffer from a severe form of myasthenia gravis, as they have this severe bulbar dysfunction and respiratory insufficiency. They have also severe weakness of the facial, neck, tongue, and muscles, And in contrast to the AChR myasthenia gravis, alterations in the thymus are rarely occurred, so thymectomy is not a good treatment for them. And we heard a lot about treatments modalities employed in these patients, and the mainstay of treatment is the use of corticosteroids and rituximab, which is a B-cell depleted agent. So when I come to the lab of Sarah and Miri, that we mainly focused on the AChR myasthenia experimental model, we wanted to test new modalities in the MuSK myasthenia, but for that, I first needed to establish a MuSK experimental model. So we wanted to induce the disease by immunization with a recombinant MuSK protein, and for that, we needed first to produce it. So we used this plasmid that was kindly provided to us by Anna Punga and Filippo Oliveri. And this plasmid contained an insert that is the histag extracellular domain recombinant rat MuSK. So we transformed bacteria with the plasmid and later on transfected mammalian cells for the production of this protein that was secreted to the medium. And we isolated and purified it, and we got 95% of purity. But for the large-scale production of this protein, we collaborated with the proteomic unit at the Weizmann Institute. So now that we had this antigen available to us to induce the model, we followed the protocol that was published in 2012 by Mori, where he showed that the FVBN strain mice, which are C5 complement deficient, are highly susceptible to the induction of MuSK EAMG. So we injected the mice with the recombinant protein emulsified in complete round adjuvant at the hind foot pads, 6 well site-separated site in the back, and also in the base of the tail. We boosted all mice 2 weeks later, but this time only on the back and with incomplete round adjuvant. Control mice received PBS emulsified in CFA or IFA only. We monitored the mice for well-being and clinical symptoms, and we gave a score, as you can see here, according to the clinical symptoms that appeared, such as tremors, severe muscle weakness, and so on. So as you can see here in graph A, all MuSK immunized mice manifested the clinical symptoms of disease, including severe muscle weakness and tremors 2 weeks after the second injection. We also observed a prominent cervicothoracic hump indicating weak of the cervical extensor muscles, and also ungroomed 4. We also monitored for the weight loss of these mice, and as you can see here, oops, how can I go back? Oh. We monitored the weight loss of these immunized mice, and we found that there was a pronounced weight loss in the MuSK immunized mice, where in the control mice, no clinical symptoms of disease were observed, as well as weight loss. We evaluated also the anti-MuSK specific total IgG antibodies present in the sera of the mice, and found that all immunized MuSK mice had detectable antibody levels. And in contrast to the AChR model, where there is no correlation between the antibody titer and the disease severity, in our case, it seems to be a good correlation between the antibody titers and disease severity, and such correlation was also observed and reported in MuSK myasthenia gravis patients. Next, we wanted to know if MuSK immunization resulted in a specific muscle damage, and for that we isolated RNA from the masseter muscles of our mice and performed qPCR analysis and found the cathepsin L levels to be highly elevated in the MuSK immunized mice, suggesting that there is damage in the muscle of these mice, as cathepsin L is an lysosomal endopeptidase enzyme that is involved in the initiation of degradation of proteins. We also saw that the levels of IL-15 were decreased in the MuSK immunized mice, and this cytokine is highly expressed in skeletal muscles, and it's believed to be a myokine that improves glucose homeostasis and oxidative metabolism. Interestingly, we saw that the MuSK expression levels were elevated in our immunized mice, and this could be explained maybe by a compensatory mechanism. Next, we evaluated subpopulation of T cells from the spleen, and after isolating the cells and subjecting them to flow cytometry analysis, we found that the population corresponding to the T regulatory cells, which are defined as CD4, CD25 FOXP3 positive cells, where the frequency of these cells were significantly decreased in our MuSK immunized mice, suggesting that there might be some alterations in this population. We further confirmed our observations by performing qPCR analysis and found the expression levels of FOXP3 to be reduced. This was also accompanied by higher elevation expression levels of the pro-inflammatory cytokine IL-18, which is related to a Th1 response. So we now have this established model for MuSK myasthenia gravis. So the next goal was to try new treatment modalities, and we chose to induce oral tolerance induction. And oral tolerance is an active process of local and systemic immune unresponsiveness to orally ingested antigens, such as food. The gut immune system must balance its responses to commensal bacteria or harmful antigens and pathogens in order to maintain this homeostasis. And this is reached specifically by specialized population of immune cells and lymph nodes that create this specific environment in the gut. We also know that there are systemic effector sites, such as the liver, that are also critical for maintaining and establishing this oral tolerance. So how does the oral tolerance work? Well, there are many mechanisms, but one of the prime determinants is the dose of the antigen fed. We know that high dose of antigens results in anergy or deletion of T effector cells, and this is due to conjugation between the T effector cells with an antigen presenting cell, but the immunologic synapse that is formed is defect, resulting in this unresponsiveness. We also know that low administration of antigen results in the induction of T regulatory cells, and this process is mainly related to the gut dendritic cells. Specifically, we know that mucosal dendritic cells can cause the induction of FOXP3 regulatory cells by the production of TGF-beta. And it is known that also all major subpopulation of T regulatory cells can be induced or activated in low dose tolerance. But why to choose this approach as a potential therapy in autoimmunity? Well, ideally, we talked about it a lot. In autoimmunity, we like to have this treatment that specifically delete the antigen-specific autoimmune response, but leaving the rest of the immune system intact. And oral tolerance, it is not toxic, it's easy to administer, and it has this specific, this antigen specificity, so it makes an attractive approach to prevent unwanted immune responses that can cause a variety of diseases or that can complicate treatment of ongoing disease. And the number of studies that address this approach in experimental model is quite large, as you can see here, such as colitis, diabetes, EAE, food allergies. And as you can see, also myasthenia gravis is in this large list. But when we address the number of studies in human, this list is quite limited. And this is because some issues that emerge concerning the application of this approach. For one, not always the autoantigens to be used for feeding is known, and it also requires a large amount of it, and it's not always doable. Also, it needs to be taken in consideration that there is a low efficacy of this treatment in synthesized individuals, and moreover, the age of the first administration is very critical for the susceptibility to the treatment. There were several studies in EAMG that wanted to prevent the disease by oral or nasal administration of the torpedo AChR that had a great success in suppressing the induction of the disease and also the suppression of humoral and cellular responses to the AChR. But here I'm showing you a study that was performed by Im Sing Ya, that was a former lab member of our lab, and he treated in the ACHR model rats with the human alpha 1, 2, and 205 recombinant fragment that correspond to the extracellular domain of the ACHR. And not only that he succeeded to prevent the induction of the disease, as you can see, in here, but he also succeeded in suppressing already an ongoing disease when he started the feeding at the chronic phase of the disease, which is an established disease. And the mechanism underlying his therapeutic effect that he got is mainly due to a shift between a Th1 response towards a T regulatory response. So we were very we wanted to test this approach in our established model for MuSK myasthenia gravis, and for that we fed our FBBN mice with the recombinant MuSK or with an irrelevant protein, which was the ovalbumin. We started the feeding 1 week before disease induction, 3 times a week through the whole experiment. And we were very excited to see that the MuSK-fed mice manifested the very ameliorated disease as compared to the ova-fed mice. And this was also accompanied by a less pronounced body weight loss. Also, the antibody titers present in the sera of the MuSK-fed mice were significantly lower as compared to the ova-fed mice. And last, we wanted to evaluate some genes in the spleens of the mice by qPCR analysis, and we found that the expression levels of FOXP3 and TGF-beta were significantly elevated in the MuSK-fed mice, and which are crucial for induction and maintenance of T regulatory cells. And also the levels of IL-10 were significantly decreased, suggesting that oral tolerance caused this therapeutic effect by induction of T regulatory cells rather than anergy. So to conclude my talk, I have showed you that immunization of FBBN mice with a recombinant rat MuSK resulted in a very reliable model for the study of MuSK myasthenia gravis that resembles well the human disease, as it has a good correlation between the antibody titers and disease severity. Also, that T regulatory cells may play a role in this immunopathology, and the administration of a recombinant MuSK protein by oral administration resulted in a significant therapeutic effect that may be further studied in order to pave the way for new treatment modalities. I would like to end by thanking Sarah Fuchs and Miri Sorojon for mentoring me during a magic time in the Advancement Institute. Also my colleague, Revita Larija, Anna and Filippo for the kind present, and our funding, and especially you for listening. Thank you very much.
Presentations are open for discussion.
Question for Professor Lazaridis. Regarding the immunization, I think you alluded to this at the end of the presentation, but did you check the subclasses that were induced?
The subclasses of antibodies that were induced in response to treatment or to immunization? I didn't get that.
Both, so like before and then after.
Okay. Yes, well, especially in response to treatment, yes, to immunization, sorry, yes, it's mostly IgG2B. In rats, the subclasses are slightly different. We have looked at the subclass distribution in response to treatment as well, but there the situation is more complicated because, first of all, as you saw in certain treatment groups, we saw an increase in antibody levels, but not in others. So in trying to understand what the nature of these antibodies is, one of the first things we did was subclasses. We didn't see it. Basically, it depends on the time of when treatment is administered. Later time point treatment does not seem to affect the subclass distribution, whereas earlier time point treatments change the balance of the subclasses. But I'm not sure yet because of the overall increase in antibodies that we see, I'm not sure whether there's something masking the details especially in the subclasses that are in lower concentrations to begin with.
In the experimental mouse PV model, we would do a small amount weekly for 6 weeks, and that induces an IgG1 dominant response, which is similar to human IgG4. And when we were doing the MuSK EAMG model, when we did a day 0, day 21 prime and boost, it would be 50-50 with IgG1 and IgG2b. And if we did weekly times 6, we would get an IgG1 predominant response, but it would go up and it would go away spontaneously. So I think, definitely, I mean, that is sort of the protocol that induces tolerance usually. So I suspect it may be IL-10, switch to IgG1, with a chronic, and then, induction of Tregs or something along those lines. But it's great work. Thanks.
Thank you.
Yeah, sorry, I was just checking for a moment. I was just going to ask about subclasses, but of course the mouse subclasses are different from the human subclasses, but that applies more to the MuSK immunization.
I'm sorry.
No, okay, well, we can leave that. I'll just continue with Konstantinos. I missed the very beginning, so I'm not quite sure, but the ECD is how much of the alpha subunit extracellular domain, the whole thing, and is it
roughly
structured?
the first 210 amino acids.
And what sort of structure is it in?
So the protein that we use is expressed in yeast, and it has what we say near-native conformation. So we checked it with monoclonal and MG-SERA antibody binding, and it's indistinguishable for the majority of samples from wild type from the receptor. So we think it has very near-native conformation. It can't be identical for a number of reasons, but it has very good conformation, the protein that's expressed in yeast.
And did you choose that in preference to something that was less conformational, which might also be able to affect, for instance, T cell responses to the same sequences?
So initially we chose that simply because it's a tool that we have very easily available in the lab. And it works very nicely, and it's also it's very easy to handle, so it gives very reliable data. There's other protein from other sources that we have might be more prone to degradation or to aggregation and so on. So that was one reason. But eventually we did try, and we did use protein from, for example, E. coli to see if it makes a difference, if the treatment efficiency is the same.
And you don't think it's acting as some sort of decoy for antibodies? For
the antibodies? No, that might contribute very short term though to the effect. We have no way of excluding that, but if it did, it would be very short term. And overall, I don't think, I mean, in the past, for example, we've done apheresis studies in the same animal model, and we see that when we remove the autoantibodies, there is an improvement. But then if you stop treatment, the antibodies and the symptoms come back within a matter of 3 to 5 days, something like that. And here the effect was lasting much longer. And also, if we look at the amounts, the relative amounts and concentrations of the antibodies and the protein, I don't think it plays a major role in the effect that we observe.
Sorry if I'm going on, but if it's segregate, no, I can't think of the right words. If it actually deposits in the liver mostly, would that not also act as a decoy, perhaps, and destroy antibodies and your ECD?
Sure. Okay. Let's say it does, but then treatment is terminated. Treatment is applied only for 12 days. Okay. After that, there's no more injections, but the disease does not come back, and the symptoms continue to improve depending on what time window we're looking at.
Okay, but
so even if that were the case, there's definitely something else going on as well.
Maybe that's the cause of the improvement. But anyway, thank you.
Thank you.
Yes, Professor. Question for you. about the anti-rat AChR response. I mean, did you quantify it as one question, and did you look at the muscle itself by immunofluorescence to see this antibodies really bind and activate complement?
No, we haven't.
And what about the titer? What's the anti-rat titer? I mean, I know you can in nanomolar.
What the actual titer is?
Yeah,
I don't remember it, but it's significantly. So we're talking again after treatment.
yes, you show the peak in anti-rat antibodies.
I don't remember the actual data, and there is quite a bit of variability there. So if you saw, the error bars were quite huge there because there's variability between the animals, but it's a significant increase compared to, let's say, untreated.
Yeah, but the absolute number might be important. I mean, if you immunize with torpedo, about 0.5% of your anti-torpedo response cross-reacts with rat, but then you're in the nanomolar range, the same as you find in patients. But what you have here probably might be a small peak that is probably not relevant.
No. So roughly it is in the nanomolar range, and so let's say in PBS rats, it's anywhere from a few nanomolar to a few dozen nanomolar, depending on also when you look during disease progression. But after treatment, it was up to the dozens or hundreds of nanomolar. Okay, depending again on when what timing we were looking at.
Because if you want to apply that to human beings, of course, for regulatory agency, it might be a bit scaring that you show that your immune response goes up against the self-antigen, and you're immunizing with human receptor, and you have a significant cross-reaction with the rat. Of course, in humans, you're immunized you tolerize with human, but you can also immunize with human receptor, and so that might actually exacerbate the disease. So that's a bit worrisome. Or you think so?
But that actually, there is some in PBS or in non-treated rats, there is some weak correlation between the antibody titers and the score or the disease severity, especially when we're looking at the AChR antibodies. ECD, not so much. But that correlation is completely obliterated when we study those groups. So there's clearly something in the nature of those antibodies that we don't understand yet. They're not appear to be linked to pathogenicity, but unfortunately, I can't say anymore. This is what we've seen so far, but we haven't characterized them any further, and that's what we absolutely need to do to see what these antibodies are.
Because in other experiments I did in Lindström's lab, I did experiments with reduced and carboxyl-methylated receptor. Actually, was a protocol made by Sarah Fuchs, and what happened then is there you get an anti-rat response, but it's mainly against cytoplasmic domains. Of course, here you only have the ECD, so that's probably not an explanation that, because you can have antibody against cytoplasmic domains and they are not pathogenic. So you find in the serum, because in your radioimmunoassay using soluble receptor, and you don't. So you don't think that in this case the specificity might be different?
Well, the epitope specificity might be different, but I doubt that it's solely because of intracellular, for example, domains or sequences, because there was also a spike in the ECD, anti-ECD antibodies.
Okay, thank you.
Okay. We have another question.
Yeah, regarding oral tolerance, I assume that anybody who eats meat is constantly getting a pretty good tolerogenic dose of all muscle proteins. So would you expect it to be protected?
Can you repeat the question, please? I'm sorry,
Yes,
I'm quite nervous.
Sure, no so regarding oral tolerance,
Yes.
I guess just that anyone who eats meat, who eats animal muscles,
Yes.
must be getting a tolerogenic dose of muscle proteins, including MuSK and acetylcholine receptor. Would you expect it to be tolerogenic, or?
I don't know how to answer this question, but we got this pronounced phenotype in these mice even after the experiment had ended, and we stopped feeding the mice. The control mice died, and the MuSK immunized mice, MuSK fed mice still, as you said, the disease didn't come back, right. And they looked normal, and so I guess that that the dose of the antigen fed, it affects if you will develop a tolerogenic response.
What is your dose? How much do you give to the rats?
We gave 120 micrograms per dose, 3 times a week for 6 weeks.
Thank you.
Just relating to that, I'm not sure if anybody's done a study on vegetarians, myasthenia in vegetarians.
No, I don't believe so.
What I wanted to ask you, Debbie, was about the subclasses of your antibodies. Have you looked at those? And can you remind us, somebody, of the relationship between the mouse subclasses and the human ones? Because IgG4 in humans is rather special. I think there is an equivalent in mice, but it's not monovalent as far as I know.
It is a good question, but unfortunately, we only check the total IgG anti-MuSK specific antibodies. We didn't do that, but yes, this is something that needs to be done.
Because if those antibodies are divalent, then all the work that's been done in the humans, myasthenia, with suggesting that it's the monovalent antibodies that are most pathogenic.
Yes,
would probably not be reproduced, and that's quite an interesting question. Perhaps you could make some of them monovalent.
Definitely.
The only other thing I wanted to ask you about was, you said right at the beginning that you looked for muscle damage, as it were. So where do you think that muscle damage, the IL-15 upregulation and cathepsin-1, where do you think it's occurring, and have you tried to find out?
No, we didn't. As we saw that these mice they have this severe weakness and they were unable to move. And I think I didn't know if the weight loss that we found is due to the reason that they cannot could reach the food, or because something is wrong with chewing. And that was the reason that we took the masseter muscles in order to verify it, and also because we know that in MuSK myasthenia gravis, they suffer from in these muscles. So this was the reason that we check IL-15 in order to answer this question.
It'd be interesting to know if it's upregulated at the neuromuscular junctions, for instance. I mean, that would be an obvious thing to look at. Thank you. And thanks both of you for very, very nice, clear presentations.
Thank you very much.
And also would be interesting to look for mitochondrial methylation, for example, that had been described. So we are finished now. So thank you so much for your attention. And this session is closed. And Carolina.
I'm running before you all go. I just have a few announcements. The first one has to do with food. So you'll want to listen to this. So I hear there's a chance of rain tomorrow, unfortunately. So meals will be served in a different place at the Riverfront South, which is upstairs at the lobby level, I think right above us. So that's one. Two, remember that the MGFA has a scientific session during ANEM that will be held on September 21st. And the abstract site is open for anyone who wants to submit an abstract for that session. Is closing on June 15, and this meeting is in Nashville, Tennessee, that I've heard is a really nice place to visit too. So you're all welcome to send your signs there. Third, if you look at the program, there's another session coming in about 15 minutes. It's a special session from MGNet. The program says outcome measure training, but I've been told it's not a training session. So this is not to train people for studies. Rather, MGNet has had a working group on outcome measures for myasthenia, trying to standardize measures, because we've seen that there's differences in how people interpret. Sometimes the instructions have not been clear or standardized. So they're going to present the result of the work and some of the surveys. So, and I think they want input from all of you. And I understand that Amanda Guidon is also going to be talking about telehealth and standardization of telehealth assessments for myasthenia. So you're all welcome. You don't need to be in an MGNet program. You're all welcome to attend. And finally, remember, we have our posters at 6 o'clock. They're different than yesterday. We have the other half of the posters. So please come back and look at the posters. And again, thank you all for coming. Thank you for everyone who's seeing us virtually, and for all the presenters for all the great talks. I'll see you later.